this approach to visualize this hidden activity? Well, in order to do that, we have to use two simple tricks. And the first one is very easy to guess. First of all, we have to get rid of this visible activity or minimize it. And that's very simple. What you do, you ask your subject to lie in the scanner, awake, still, and you don't present any stimulus to them. So you image them at rest. Simple. The second trick is a little bit more subtle. And it is more subtle because it has to do with the very nature of this phenomenon, which is a subtle phenomenon. So let me explain with an example. So what you see here on the top is the signal I just showed you. Okay, so this is a sort of visible activity that it is elicited, triggered by stimuli. By contrast, this is the sort of signal we can record when your subject is at rest, as I told you before, no stimulus. And as you can see, nothing much is happening, right? There are these squiggles and that's it. But look what happens if I extend my recording to 1 minute and to 2 minutes. See what's happening? A very subtle sort of rhythm, this wave, becomes apparent. And this is a crucial bit of information. This suggests that beneath this noisy resting signal lies a symphony of activity that we've been totally overlooking. And so this led scientists to think that this activity here could be, in fact, a manifestation of this elusive dark energy we've been mentioning, I mentioned before. And just to visualize it, we just needed to suppress this elicited visible activity, but also to scan for longer, so simple. And there you have it. This is the sort of signal we can map with the approaches I show you. No stimulation, resting activity. And what I find fascinating about this sort of activity is that you can learn a lot about this phenomenon just by simply watching this video. You don't need to have a PhD in neuroscience here. The first thing we can learn is that, as you can see, your brain never rests. And even if you are asleep or if you're not doing anything, this activity is on 24/7. And this explains why this sort of activity is so metabolically costly, okay? It's expensive, always on. The second interesting fact here, you can see very well that this activity is not noise, but it is in fact structured into slow waves that propagate across the brain. And the fact that this activity is so slow, it's a reason why it has eluded us for so long. These waves are 100 times slower than typical brain pulses that neuroscientists have been measuring over the years. And the third fact that I like to emphasize here is that this activity is also synchronously rising and falling across the two hemispheres, the two sides of the brain. And this is really crucial because this reflects the brain's ability to coordinate the activity of the different brain regions, basically, very much like a conductor leading a symphony. And brain coordination is vital for effective and healthy brain function. And so this synchronization, this sort of coordination, is at the basis of some sort of like a novel brain mapping approach that we can now implement based on these measurements. We can transform these signals with very simple mathematical tricks into these sort of images, which I call here for simplicity brain synchrony maps. Interpreting these images is very simple. Color here indicates how synchronous signals are. So regions that have the same color, they play the same melody. Different colors are different melodies as part of this symphony of activity, okay? And so by doing this is very simple. We can go beyond anatomy. We can now probe and test how harmonious these waves are, how synchronous they are. But we can also map their volume, how loud they are at the whole brain level. And this new insight has led to groundbreaking discoveries. And for one I want to mention, which I find very fascinating, is that this dark energy, this hidden activity, is at the basis of who we are as individuals. How do we know that? Well, if you compare the brain synchrony maps across individuals, you can see that their structure is pretty much the same, right? But there are subtle variations. And these subtle variations across maps create unique, reproducible brain fingerprints, where every subject has got this unique, distinctive pattern. And so these synchrony patterns are related to who we are, and of course they're not fixed, they change. We're learning, they become more mature and they evolve as we go from childhood into adulthood, and they kind of become dissonant and they decline a little as we age. But one of the most interesting applications of this novel mapping approach comes from its use to study brain disorders. And here, pretty much all the research that has been carried out so far point in the same direction. All brain disorders, whether they are psychiatric, neurological, developmental, disrupt or alter this sort of synchrony maps. And this, per se, is already an important finding that suggests that this sort of mechanism, this phenomenon, is at the basis of healthy brain functioning. If it's disrupted, we cannot function properly. But there's more. By studying how these synchrony maps are altered in brain disorders, we were kind of forced to completely rethink how we approach brain disease. And I want to show you an example of what I mean. Now, in my lab, we've been studying how autism affects these brain synchrony maps as measured with MRI. And so what we did was something very simple. We used like a public large database of MRI scans containing like more than 100 autistic individuals. For each individual, we calculated a synchrony map, and then we compared each map with an average synchrony map for control population, typically developing people. And by doing that, we can get like difference maps that tell you where and how this symphony of signal is altered. And with this simple approach, we learned a lot. This was really enlightening. And the main take-home messages are two, I guess. The first one, perhaps as postulated, as we expected, the vast majority of autistic people have major alterations in this sort of synchrony maps. But what is really astonishing is the huge variety, the huge variability that we found across maps within autism. So these maps across people with autism are very different. Let me show you what I mean. We found that some people show under synchrony, which means that this symphony is out of tune, it's dissonant. But some other autistic people show the exact opposite. They show over synchrony, which means that their brain orchestra plays the same tune over and over. There's no variability. And some other people show a little bit of both, basically. That's, under and over synchrony. Yet all these individuals have been diagnosed with autism. And so the question here is, why so much variability? What does this variability encode for? Well, one hypothesis we've been exploring in my lab is that, in fact, there is no one thing or such thing as one autism, but there are many biologically distinct autisms. And we also hypothesize that we can tell these apart using this sort of signal. However, rigorously testing this hypothesis is difficult because we cannot, at present, measuring biological dysfunction in the living human brain. And so we thought, okay, if this cannot be done in humans, maybe we can use like a model organism, like the laboratory mouse, which is what we've been doing. And while this may seem like a bit of a crazy or unconventional idea, it actually makes a lot of sense. And it makes sense because, first of all, this symphony of signals is evolutionarily conserved. You can map it also in monkeys or rodents. Clearly, in rodents, it's more primitive, it's simpler, right? These melodies are very simple. But they are evolutionarily conserved, so their structure is conserved enough to allow some translation across species with some caveats. The second important point is that if you move to rodents, all the mice we use are genetically identical except for the mechanism we want to study. And this is a key advantage because by imaging them, we can get rid of this individual variability. We can really isolate the effect of the mechanism we want to study. And so we gave it a go. Over the past 10 years or so, we mapped this MRI synchrony, these synchrony maps, over more than 20 mouse lines, each modeling a single genetic and/or immune-related mechanism that is important for autism. And by doing that, we were able to recreate the biological diversity of autism, right? And the results we got are represented here. And these are brain synchrony alteration maps produced by each of these biological factors we tested. And if I add this overlay, I'm sure it rings a bell, right? Looks familiar. This is mirroring what I showed you before in humans. Some mechanisms, some alterations produce under synchrony, some other produce over synchrony, some other produce a mix of both. But there's more. Now that we know that these mechanisms, we can also tell that these blue ones, so under synchrony, is strongly associated with alterations in our synapses, with our neurons, and over synchrony associated mostly with immune-related alterations. And so this is a very important result. First of all, it explains why there is so much variability in the maps we map in autistic people. Biological diversity reflects or results actually in different forms of brain dysfunction. And this result also suggests that this under synchrony and over synchrony might indeed lead to different dissociable forms of autism. But these are mice. Mice are not humans. We all know that, right? And I agree. So in order to test whether this works also applies to humans, we went back to our human database with all the scans of autistic people, and this time we selected out only those that have under or over synchrony in the same regions we map in the mouse. So we use the mouse as a prior, as a guide. And by doing something so simple, we were able to find that an astonishing 40% of human autistic cases fall into these two categories. So match the same under or over synchrony we found in rodents. But that's more. Regions that show under synchrony or over synchrony humans are spatially enriched with the same synaptic genes we found and we modeled in rodents, or immune-related genes that we modeled in rodents, depending on the two groups. And once again, this closes the loop. This shows basically that now with this approach, we can decode these dark energy maps into their biological determinants. So we can now link these patterns of altered symphony activity with their biology. And so in conclusion, our journey into the mysteries of the brain's dark energy led us to realize how this phenomenon is at the basis of who we are as individuals and how it shapes our cognition, our behavior, and mental health. And by decoding the biology behind these maps, we hope to chart a future where precision medicine will hopefully provide relief to the many that still suffer from brain disease. And what's more exciting, as far as I'm concerned, is that this journey has just begun. Thank you.
Thank you very much, Alessandro, for this fantastic journey. And Masha, I don't know if we are, I was going with the streaming, and if there are questions, if we can add also a question session to this
video. We still have some few minutes if you want, so he might answer to some
somebody
questions.
of you has question for Alessandro. The first, David. I can.
Okay, you can give the microphone.
This is going to be embarrassing.
You hear me?
Yeah.
Yeah, I was wondering because indeed, I think first of all, really nice presentation and larger work, and we haven't presented it. Second of all, I was wondering indeed you are correlating basically the subpopulations of autism with their biological signatures. And I was wondering if indeed also using the database, for example, the open database that you were using for, if you can also relate it to some phenotypical types, because of course autism is a spectrum, so it could be more closer to ADHD or to intellectual disabilities. So
Yeah, no, that's a question that makes a lot of sense. Yeah, many people are doing that, and this is really part of like a larger effort that is ongoing both in our institute with other investigators, but also throughout many other institutes in the world. I think that the problem here is to have like, if you want to partition complex disorders such as autism, we for sure need to use like a multidimensional space where phenotypes like behavior is one of these dimensions. The trouble that we've been having so far is the poor correlation between behavior and some biologies. And this is a novel dimension that we can follow. And without getting too technical, clearly here is tons of details, and it was like, higher level. But I think what we show here is proof of concept that, you have to go a little bit beyond like basic machine learning or sort of like clustering. Of course, you can cluster everything, right? You can cluster you can cluster whatever you want. But here we provide some, sort of like ground truth biological validation to tell or to guide this algorithm and say, okay, this group is like this sort of dysfunction, this other group is this other dysfunction. And so that's already, an advancement towards the state of the art, which is like, yeah, let's cluster and let's hope that we can classify.
There was another hand raised before.
I think I didn't understand how under and over synchrony is linked with synaptic genes and immune-related genes.
You did not understand how? If there was a question? Yeah, I mean, maybe you can go back quickly. It's very simple, and this could be done very, very easily in the mouse. If you put together, you see these are like difference maps for each mutation. So you've got a tag here. This one is SHANK3, it's a gene we know in synapses. This one is 16p11.2, which is the chromosomal deletion. And then what we did there, we created these gene lists. We removed all the genes that are in common between two, because of course there's some share, and then we just kept the list that are specific to blue or to red. And if you do that, you get this massive enrichment. You can really separate these two. If you see what I mean, is it clear? So that's the advantage of using the mouse, basically, because you have this hypothesis that you can test.
So, thank you very much for this presentation, really wonderful. I was wondering, these synchrony maps, do you know anything about their development? And, most of the work I think was more adult, and I was wondering, what about childhood?
Well, yeah, this is another very interesting and relevant question. Well, I think it's what you said is partly incorrect, and clearly I don't blame you because it's been explained, but a lot of this work is in fact done, especially in humans. These maps are acquired in like children. But as I mentioned in passing, these maps evolve quite stereotypically over development. At the beginning, for example, what you see when young children, you have like this synchrony is only on sensory regions, and all this integration, like prefrontal regions more evolved, they become synchronous. So melodies there, appear later, and so you have this maturation where you have these complex patterns that happen like later in life. And so this is interesting because if you just project some of this stuff here on the evolutionary scale, you can, this is really like tentative, but interesting. You can maybe say that some of these patterns are like, a reflection of a less mature brain, so to speak, right? So it's like you can see a deflection of the maturation. But yeah, the evolution is pretty remarkable, and it is an important variable to consider.
Thank you for your presentation. I got a crazy question. I know here in Pisa, there's a professor called Giuliani and Neri. They're doing some research about the brainwave of Tibetan monks when they are doing meditation. Like what they found out is that the Tibetan monks, when they do a specific meditation called meditation of concentration, their brainwave is in a state between the wave and sleep. I don't know if you, in the future, you are planning to do the same research.
Absolutely, this is not crazy, and people have done this already. And so, yeah, far from being crazy, absolutely. No, that's exactly one of the, what is interesting here is that in this sort of like activity, and there's also research we've been doing in the lab, what is really interesting is that these waves are really capturing the very slow leftmost part of the EEG, so to speak. Really, this frequency is below 0.1 hertz, below. Okay, that's the upper bound. And so these slow waves, for many people, have been neglected also in EEG, right? In EEG, unless you measure DC signal, you don't see that, you discard. 1 to 4, it's delta. And yeah, this sort of like slow activity can be entrained, for example, by respiration, and it is really tightly linked also with our bodily reaction. So, and that's yet another bit, but maybe it's another talk in 5 years' time, because that's yet another angle.
I was wondering how specific is oversynchrony and unsynchrony for autism? Do you see it also sometimes in people without symptoms of depression or schizophrenia?
It's not exactly, very, very good question. It's not specific to autism at all. These are just general alterations that we can decode now well. And yeah, that's exactly one of the points. People have been, and in other talks I do, like a little bit more, with like deeper in the science, I give this talk with a different angle. The angle that I start, which is exactly your question. People have been trying to use this for years to find the elusive signature of dysfunction specific to autism. And there isn't such thing, neither in humans nor in models. These are general biomarkers, so to speak. And we know more. Blue, under synchrony, we have studies in the lab, are an expression of increased excitability, cortical excitability. And the other way around, red means the cortex is more silenced. So that's really, to me, the biggest advantage. We can just decode this in terms of signal, but no specific, very little specificity, let's say.
Thank you very much. Very nice work. I was wondering if you try to use this in any therapeutic context, you try to repeat recordings under any of the therapies that may be available in conference and methodology that you were trying to how they would get involved in that.
I'm afraid I didn't catch exactly the question because there's like an echo. If I use this to do what exactly?
In any therapeutic context.
Therapeutic, yes.
Yeah.
Yeah. Well, as I mentioned before, what we are now pursuing as part with big clinical efforts is to use this, especially the human subtypes, to guide, therapy for autism is a little controversial. We have to be very careful because clearly not all autism, right, diagnosis require therapy, but there are, forms that are strongly associated with like disability. But the idea here is that we can tell apart the two wings of the spectrum. And what we are proposing here, we have to get this published soon, by the way, is that blue under synchrony is a marker, a bona fide marker of increased overly excitation of the cortex. And we have chemogenetic manipulations, a lot of studies that I couldn't show today that support this. So the idea is that by using this, we should be able to find people that have like excessive excitability in the cortex and the other way around. And of course, if you want to go for treatment, at the very least, we have to go, with diverging sort of options with these two populations. So yes, it was like elaborate response for yes.
Hi, thank you very much for the talk. So as you just said that it's not specific for autism, you can see it in schizophrenia and stuff. So I was wondering, do you think that the undersynchrony and oversynchrony are a state-dependent thing, or is it maybe a type-dependent trait?
They are a bit of both, I'm afraid, as often the case in neuroscience, right? They are definitely state-dependent. Arousal increases, decreases synchronization. Well, it's a little bit like EEG, right? We are not really reinventing anything here. Many people, think of this like a specially resolved, very slow EEG, okay? And so, very much like with our EEG, when we are asleep, we have all these synchronous waves, right? So if you are like drowsy in the scanner, or like low arousal, you will have a little bit like more synchrony. And the other way around, if you are highly aroused, you see a desynchronization. And this is really a stark contrast we see in mice. I'm sure these days you discuss a lot of mouse work, also wake and fix, this sort of stuff. We did report, we published this symphony in awake mice, and those guys, even if you train them, they're happy to be there because they don't do well, relatively happy. Yeah, I mean, they are always super aroused, right? They never, and you know what mice are like, right? They're really these crazy fellows that do, and they never keep still. And so you see, if you compare with light sedation, you see that these maps are strongly synchronized. So yes, centrally arousing signals play a role. And it's a little complex. Maybe we can discuss offline because, yeah.
What is the effect? Is there any study the effect of environmental conditions, such as part of electric pollution, on the behavior of this synchronicity?
No, that I know of. No, that's not my field. So I cannot rule it out because there are many studies. This is a very simple approach, but no, that's not something I know. So there might be, but I'm not aware of that.
Yeah, thanks for the visualization of the fMRI. That was really good. But also with that, like I've seen real fMRI data, and like you don't see anything. So already the pictures that you showed, like it contains a lot of reconstruction. And so I was wondering, like when you look at like mice data, just like the brain, like the precision is like much higher when it comes to like temporal and spatial resolution. And so once you start clustering them, like did you encounter any issues like to put them together, And also,
I confess, I had trouble in. So you're asking about. Give it another go. I'm sorry. I wake up very early this morning and. Yeah.
The temporal resolution of fMRI is 2 seconds, right?
Yeah, it's very slow. Yeah.
And temporal resolution of calcium imaging, for instance, is like 40 minutes.
Yes.
If you then do like clustering, and also fMRI is just measuring blood, right? Like calcium is measuring the neuron activity. So just what you're measuring is very different. Once you start clustering the data, maybe it's just like surprising that you can like find still the same correlation. So like where there are some approximations you have to do to basically.
Well, I mean, just to elaborate on your point, I mean, what I can tell you for sure is this: if we had EEG for all these fellows here, we will have for sure different clusters. There's no question. Because EEG is richer, and there are also these higher frequency activities, right? No question. MRI, this signal here, is sensitive to the very leftmost part of the EEG. And this is stuff that we have proven in the lab. We haven't published yet, but we're about to publish soon with manipulations. So calcium, which is a little faster, not that faster, but a little faster, or EEG, can produce much more variety. That's why I said before, I'm not proposing that this is the only categorization of autism. That would be just insane. This is one of the dimensions. But there are lots of databases with this that now we can make use of. Before it was just a phenomenological description. Yeah, there's more connectivity. This is called, by the way, functional connectivity by people, which is a very misleading term. Maybe we should drop it. This is synchrony. So that's all I'm saying, basically, okay? And that's pretty much it. And another thing I can add is that if one day we let AQ these models, probably we find more categories. Maybe the blue we can split. As you can see, in immune-related, it's not only immune-related, there are FNR1 and other stuff that we can maybe separate. But yeah, so far, so good.
One last very quick question. If like schizophrenia and depression, some motivation show that they are similar to some similarities between them, like are we getting over the normal biological categories?
I think not based on this data set per se, but I think we should and must go beyond this very, like face validity sort of classification. There's consensus in the field. I'm an imaging guy, But I'm in close contact with many clinicians, and I think the consensus is emerging that we really have to go beyond that. And once again, I'm not saying that this is the only key, right? This is one of the dimensions. The space has to be, now also with AI, there's really like many ways of partitioning this complex multidimensional space. And I think this is where the field is going. There's no doubt for me.
Okay, thank you very much, Alessandro, for this fantastic talk, and thank you, you all, for coming, for raising this very interesting discussion. And I think that now we can close our ITN conference, Serotonin conference under the Green Tower. Thank you, Judith. Thank you to all the students, all the PIs, the Scientific External Advisory Board. And so I hope we will have other occasions to meet here together, talking about serotonin, ASD, and schizophrenia, depression, and enjoy science, enjoy also communication of science with